Yeah, so my name is Colton Lloyd, and I'm a fifth year in the Systems Biology Research Group. And today I'm going to be talking about models of metabolism and gene expression, or me models. I think they've probably been kind of touched on a little bit so far in the class, but today I'm going to talk more about the fundamentals, about what these models actually, how they work, basically. And then next, in a couple weeks, Lawrence is going to come in and tell you more about what some of the exciting things you can actually do with them. So the overview of today, first the first part of this is we're going to understand the motivation behind developing me models. So this is two parts. What are the limitations of M models? And then how does a me model actually come in and address some of these issues and expand what they're capable of? The second part is to understand the major underlying differences between M models and me models, and fundamentally what this means is how do you reconstruct a me model starting with an M model? And then the last part is going to be to actually understand the new me model capabilities and some of the major challenges associated with these new capabilities. So this means what new avenues does the me model allow you to study, and so on. So just as a little bit of background, I'm sure you guys are very familiar with this at this point, just generally constraint-based reconstruction and analysis. And what this method does is it takes the organism's metabolic reaction catalog and converts it into a type of computational format. So it takes pretty much the catalog of every reaction that an organism is known to be able to catalyze. And it converts this into what's called a stoichiometric matrix, kind of detailing the possible stoichiometries of the reaction the organism can accomplish. Applies constraints, and then using LP type optimization software, you can learn more about what the organism is capable of. So this method has had a lot of success in predicting the metabolic capabilities of an organism. So what are feasible growth rates, or what's a possible yield of metabolite from a certain carbon substrate, among many other things. But one limitation of this type of method is that you can say that Flux is free for an M model. And basically what this means is the only thing that's connecting the genome, up here it's a little hard to see, but connecting the genome to a reaction is what we call a Boolean gene-protein reaction rule. And basically what this says is that if the genes are not knocked out in this genome, then this reaction is free to proceed basically at any rate that's feasible in the model. So as a result of this, there's a couple things you have to do to make these models predictive. One of these is you have to apply a growth rate determined biomass objective function, which is derived from empirical measurements of that organism. And you also have to apply bounds on things like the substrate-uptake fluxes to make sure that all the growth predictions and everything are feasible to what the organism can actually do. So a consequence of this is that the solutions of the M model are non-unique. So the reason for this is that the S matrix and the constraints, it defines an underdetermined system. And so this gives rise to what's called alternative optimal solutions, which I think has probably been touched on to some extent. And so one example here is for the reaction pyruvate dehydrogenase. And what this shows is that as you move from 95% of the maximum growth rate to even the maximum growth rate, the minimum and maximum flux of that reaction, even at the maximum growth rate, is really not known. It can be a range of possible different fluxes. And this is the case for even reactions like pyruvate dehydrogenase, which is a very biologically important reaction. It shows growth defects when it's knocked out, and there's been a high evolutionary pressure to optimize this protein to the fullest extent. So as a result, this enzyme is 60 subunits. It has 4 different pretty costly cofactors that are required in order for it to run. And kind of see a picture of it up here. It's one of the most complex enzymatic systems that's known to biology. So there's a huge selection pressure on trying to to get this reaction to be as efficient as possible because it's very important for growth. And even at the maximum growth rate with an M model, we don't really know what the flux of that reaction is going to be, and it can even be 0 and grow optimally, which is not ideal. So there's a couple methods that have been developed for accounting for alternative optimal solutions. One of these is called solution space sampling, which again I think has been touched on a little bit. And the way this method works is it basically simulates the model many, many different times and tries to cover this entire solution space, which is an example of this is shown here. So in doing so, the kind of binning of all the possible fluxes of that reaction throughout the entire solution space will give you some type of indication of which reactions are most likely to occur in that organism or to be on. The second of these is called parsimonious FBA, and the way this method works is basically you optimize the model under a certain set of conditions, and then you run a second optimization to try to minimize the sum of all the fluxes throughout the network. And what the reason this method was developed is we wanted to try to mimic the cell's drive to try to optimize certain its enzymatic processes, or optimize the use of those processes. The third here is kind of what I showed on the previous slide. Is called flux variability analysis. And with this approach, you kind of go through all the reactions one by one and maximize and minimize those reactions under a certain set of conditions. And so in doing so, you get a range of all the possible fluxes under those set of constraints that reaction can occur in, and kind of get an idea on the systems level what the variability is. And then the fourth is somewhat what meat models would fall under. So these are models that actually apply the proteome capacity constraints. So I'm going to talk a little bit more about those. So first of all, why is this something we're interested in doing in the first place? So beyond actually addressing alternative optimal solutions, why are we interested in including proteome capacity constraints into metabolic models? One of these reasons is the availability of protein within the cell is just inherently very limited. This is kind of shown pretty well in this figure here, where you can see how densely packed in an actual growing organism the protein and all the and everything really is. And so as a result of this, the metabolic fluxes that are catalyzed by all those proteins is very restrictive. They're very restricted. And kind of a consequence of that is E. coli over time has had to evolve to optimize its use and the way it allocates protein as much as possible in order to grow the most robustly under a wide range of conditions. So you can say while the reaction catalog or an M model defines the metabolic capabilities of the organism, the actual proteome allocation and the way it uses its proteins really defines its growth capabilities, or how effective it is at growing, or how it actually grows. The second reason we're interested in studying proteome capacity straits and the way they affect networks is this has implications for many physiological phenomena. And one example here is overflow metabolism in E. coli, or it's called the Warburg effect in cancer cells. And basically what we see is as E. coli here reaches a certain growth rate, it starts secreting acetate in order to kind of maximize its growth rate. And so this, it looks to be wasteful secretion of acetate that could be used to produce more ATP for the cell. But what's actually happening is that this is occurring as a result of cellular attempts to try to optimize the way it uses its protein. So again, this is kind of hard to see here, but fermentation pathways, while they're a little bit wasteful, they secrete acetate. The proteins that generally catalyze those reactions are very small, so they're efficient in that way. Respiration, on the other hand, it's efficient in the sense that it fully oxidizes glucose to CO2 and creates a lot of ATP. There's a lot of protein investment in actually running a lot of those processes. So beyond me models, there's a couple approaches in the past that have been used to try to model the protein capacity constraints at a systems level. And usually the way this works is they usually define some type of way of gauging the way that the protein is implemented in the system, which is shown here, some way of determining the enzymatic cost through multiple different methods. And then they either use constraints to try to minimize the protein expression throughout the network, or constrain it to some upper limit, which would be, it's a little difficult to see there. So while many of these different methods can reproduce phenomenological observations, they really cannot account for protein expression in mechanistic detail the way that's necessary to fully understand the system. So this brings us to part 2, to actually start defining what a me model is and how it's constructed from an M model. Going back to the previous illustration, where in M models, really the only thing linking the genome and the reaction is a Boolean set of gene-protein reaction rules. What me models actually do is they take this relationship and they actually add all the reactions that involved in transcription of all the RNA and then translation of all the protein and ultimately formation of the complexes that catalyze each of the individual reactions. So this makes me models multi-scale in nature. So you have these processes like transcription and translation happening on drastically different timescales. And so as a result of this, we need a way to couple each of these layers to each other. And I'm going to talk about those a little bit in a minute. And ultimately, all these macromolecules have to be produced at a certain amount in order to efficiently catalyze whatever process they're actually catalyzing. And like I said, this is done through what's called in the me model coupling constraints, and I'm going to talk a little bit more about those. So a typical in-model reaction, using kind of a economics type analogy, a typical in-model reaction considers the capital cost of running the reaction, but not the operational costs. So for each reaction that's happening, you have a sense of the cost of that reaction based on the availability or the flux available to the metabolites involved in the reaction. But you don't really have any costs of any type of indication of the cost of actually operating that reaction, so the cost of making the machinery required to make this reaction run. In cellular systems, this could be done by just adding the enzymes directly to this reaction. So since an enzyme by definition is not created or destroyed, you have it showing up in both the products, or the reactants and the products. But if you're going to do this into an M model and just apply all the enzymes in this way, you're going to have a regeneration of all the enzymes. So what you would end up seeing is there's no way for the model to actually produce the enzymes required to run that reaction. And so this is where coupling constraints actually come in. So this really at its heart is the key difference between M models and me models, is the use of all these macromolecules and the way we implement them as ways to couple different major cellular processes together. And the way this is generally done is by considering the dilution of all of the different enzymes to its daughter cell. So as a strain or some type of organism is growing faster and faster, it's going to be actually diluting all the ribosomes and major macromolecules to its daughter cells. So those are going to become lost as it grows, and you're going to have to synthesize more of those to make up for the dilution or the depletion of those macromolecules. So now I'm going to first talk about the kind of the easiest case of formulating a coupling constraint, which is for metabolic reactions, and it's coupled through the dilution of its complex. So this represents pretty much the simplest case, where the dilution of a complex to catalyze a metabolic reaction is defined by the growth rate divided by the K effective times the rate that it's used, which is kind of a depiction of how that's actually implemented right here. Basically what this means is that the enzyme is going to dilute at a rate proportional to its growth rate. So the faster it's growing, the more that's going to be diluted to its daughter cells. And the actual usage of that complex, so the rate that that enzyme's actually catalyzing different fluxes throughout the network. Then as a measure of different complexes or different proteins have different, some are more or less efficient at catalyzing certain processes. This is kind of considered based on the turnover rate of that certain enzyme. So this is kind of a pseudokinetic process to gauge how efficiently that enzyme can catalyze a certain process. Let's see. Oh, and so for a just reaction in a M model or a me model, if you're going to print the just basic string of how that reaction looks, it basically mimics exactly what you see here in this little cartoon, where you have on the right side, I think this is GAP dehydrogenase. You have on the right side here all the metabolites involved in the M model, and then this here being the complex and this being the coupling coefficient in front. So mu over K effective in this case equals this very small value times the growth rate. So basically throughout a me model you can think of the major levels that need to be coupled in terms of basically two major levels. These include coupling of protein to metabolic reactions, or kind of converting this to the model. This is coupling translation flux to metabolic flux. And this is basically exactly the example that I showed on the previous slide. So this first kind of level of coupling is already accomplished, as the turnover rate is the main difference between all the different complexes. And then the second part here you can think of as coupling the production or the synthesis of RNAs to the production of protein. So this is a case that's a little bit more complicated. It's going to be thought of as basically coupling transcription flux to translation flux. And it's calculated from a kind of well-studied nuclei growth rate-dependent RNA to protein relationship. And beyond that, just basic macromolecular properties like growth or the weight of certain components and their abundance in the cell. And so this is kind of this growth rate-dependent RNA to protein relationship that I talked about just a second ago. And so you can see in E. coli, as you grow faster, the RNA to protein ratio increases linearly. And this can be kind of defined by this simple linear relationship, where 1 over what they call KT is the slope of this line, and R naught is the y-intercept. And we're going to use that later in this example. So just as one example of how we would do a transcription to translation coupling coefficient derivation, we're going to do that here for tRNA specifically. This is just one example, but they pretty much all follow the same general approach. And so the way we would derive this or think about it is we have a rate of dilution of tRNA, and this is going to be coupled via alpha, this alpha term, to the rate that it's charged. So as the tRNA is charged, as the tRNA is charged, a certain amount of it is going to be diluted. So this is the amount that you're actually going to have to synthesize for that process to take place. And so what we're going to want to do here is we're going to want to solve for alpha, because that's the coupling coefficient. And so now we're going to look at each of these individually. So the rate of dilution here, in this case, is going to be obviously equal to the growth rate times the concentration of tRNA within the cell. So we're going to want this to be in millimoles per gram dry weight, or millimoles per gram dry weight hour, because that's the unit of flux in a COBRA model. So what we're really interested, because we're going to know the growth rate from the simulation, but what we don't know is the concentration of tRNA. And the way we calculate this is based on R here, which is a parameter here on the left, the total cellular RNA mass in grams per grams dry weight. But since this is all of the RNA, we need to know what fraction of this in E. coli is what fraction of the total RNA is tRNA. So we're going to include a parameter, it's just a constant value that represents that fraction, and then divide that by the molecular weight of an average tRNA. So this is going to give you the moles per gram dry weight, or the concentration of tRNA in that organism, or in E. coli in this case. So we have V dilution, or an expression that defines that value. And so now the only thing we need to know is V charging, or the rate that tRNA is charged. So similar to dilution, this is going to be equal to the growth rate times the concentration of incorporated amino acids. So the rate of amino acid or the concentration of amino acid that's actually incorporated into a protein or exists in a protein. And so this is going to be equal to, this concentration is going to be equal to the total protein within that cell divided by the average molecular weight of that amino acid. So this is going to give us this concentration in millimoles per gram dry weight. And so in order to get alpha, we're going to divide the dilution by the charging and solve for alpha. So what we get is R over P times these parameters from the first two steps. And since we have this growth rate dependent RNA to protein ratio that's well studied, we can replace this with this linear relationship here. And so this ultimately ends up being the coupling constraint for tRNAs as they're charged. And so this is what we're actually going to implement in the model, is this relationship here. So I'm not, obviously you need to do this for all the major macromolecule components, which are shown here. And they're all basically derived the same way as we did in the previous slide, but I'm not going to go through them individually. But all of these generally have a consistent form where we have each of the coupling coefficients being equal to mu divided by some kinetic rate of that process, similar to the K-effective for an enzymatic reaction. But basically what ends up differing is this K process term, which I showed on the previous slide. And again, for metabolic reactions, this is based on the turnover rate, so this is a measured fundamental property of that enzyme and that reaction. And then for tRNAs, rRNAs, and mRNAs, this is really amounts or ends up being a hyperbolic efficiency term that's back calculated from that fundamental RNA to protein ratio that's growth rate dependent. So this basically covers most of the coupling constraints that we're going to see in the model, and these are actually going to make up or be used to make up all the different meat model reactions. The second thing we have to do, besides defining all of the coupling coefficients and coupling constraints, is we have to rethink the way we use the biomass objective function for our meat model. So for an M model, this requires that empirically derived biomass objective function. So this is obviously necessary in order to get some type of biologically relevant prediction of cell properties. And like I said, they're determined from empirical measurements. The me model, however, this is going to dynamically compute all of the different amino acid, nucleotide, cofactor, etc. All the basically demands on that major components as a result of having to actually synthesize all those major macromolecules, the proteins and the RNAs and everything else. So obviously this is going to replace the need for most of the biomass objective function. But this still needs to be considered to some extent to make sure that basically there's a biomass is synthesized at the rate that it grows so that everything is mass balanced in the end. And so this is generally how we do that. So if you consider a cartoon of the biomass objective function for the E. coli M model, IJO1366, you have basically biomass building blocks, nucleotides, amino acids, cofactors, and then some things that aren't really included in catalytic macromolecules like cell wall components. And all of this is set to a rate that's equal to the growth rate, because that's the way the biomass objective function is defined. For the me model, however, we're going to take some of these macromolecule building blocks and replace them with RNA biomass type constraints that are actually produced in the model. And I'll show that in just a second. So instead of having nucleotides and amino acids, you have RNA biomass and protein biomass. But since cell wall components don't have an enzymatic function, they're just an inherent property of the cell or an kind of inherent characteristic, the cell wall makeup, this maintains in the me model biomass objective function. And so the way that these different biomass, in this case protein biomass constraints, are actually produced in the model is, if you consider the me model translation reaction, where you actually have a model reaction that includes amino acids or cofactors, which are catalyzed by the ribosome with its coupling constraint, which is, again, it's a little hard to see. This is going to produce protein, and then this extra constraint, this protein biomass, and this is going to have a coefficient equal to the molecular weight of that protein. And so this is the term that's actually going to be used in the me model biomass objective function. All right, so again, you can't really see it, but this is what the basic me model optimization looks like, where you're maximizing mu subject to, like the M model, the stoichiometric constraints, and also the coupling constraints, which are added as well. And so kind of a summary, or basically in the process of doing all this, it results in a full me model that is huge. It's 6 times the number of reactions and about 7 times the number of metabolites, and it's nonlinear because you actually have growth rate terms in the coefficient, or the metabolite coefficients in individual reactions. So that's going to make it nonlinear, and you can't really solve it the way you solve normal M models. So how do we simulate this? So like I said, the mu matrix is nonlinear in mu, but it is at a given mu. So if you plug a mu value in to all those coupling coefficients throughout the entire network, the me model is linear as a result of substituting that value. And also, as mu increases, the solution space shrinks, which is shown here. So for if you run FVA on a given reaction, VI, at a small growth rate, it's going to have a wide range. But as you increase the growth rate to the maximum value, this range is going to shrink to effectively 0 if it's solved with the right tolerances and everything. But additionally, it's been shown that the mu matrix is also quasi-convex. And what this means is mu greater than the maximum feasible mu, obviously, is going to be infeasible, and mu less than the optimal mu value, or growth rate value, is going to be feasible. And so as a result, we use a binary search solving algorithm, which I'm going to show on both this plot here in terms of this reaction and growth rate, and just on a normal number line. So the way this works is you start with an initial guess of mu. In this case, it's red, so it's an infeasible growth rate. It's higher than the maximum feasible growth rate. So this doesn't work, so we're going to try a number smaller. This number is very small, so it's feasible. So we're going to try a number between these two values and kind of go back and forth until we find the maximum feasible growth rate within whatever tolerance we set. So this is how these mu models are actually solved when we want to simulate with them. All right, so then the last part we're going to talk about is understanding why we did all this in the first place, what new things we can start to study with the mu model. And first I'm going to talk about kind of the properties of the me model solutions. The first property that's interesting is that the me model is self-limiting, and what this means is basically, unlike M models, the me model does not require some constraint to limit the amount of substrate uptake within the simulation. So you can basically take glucose in a simulation and leave it completely unbounded, so it can take up as much as it wants. So as a result, we get a growth curve. So this is a me model simulation curve that looks something like this. That's broken up into 3 growth regions. The first of these is the nutrient-limited region. So this is basically below the maximum growth rate, where if you give the model more glucose, it's going to grow faster in a linear rate. So basically, more glucose equals more growth. Then once we reach a glucose uptake rate that's closer to the maximum feasible growth rate, we enter what's called the Genome region, or what we call the Genome region. And here, this is where we start getting some diminishing returns. So as you increase the amount of glucose that's available to the simulation, you don't get that linear increase in growth rate, but you start getting less and less of a growth rate improvement for each unit of extra substrate. And this basically is happening because the model is starting to become proteome limited. So in this region, the model is going to behave pretty similar to an M model, but as we get to here, it's going to start making different pathway choices because different parts of the proteome is going to start being constrained. And then this third final region is what's called the batch growth region, because it's similar to a cell growing in batch growth conditions. So this is going to be basically unlimited glucose, but just like E. coli grown in the wet lab, the more glucose you give it, the faster. It doesn't continually grow faster. There's some maximum growth rate. So this is captured in this region here. So going back to this example, another thing that is very attractive with me models is each solution is going to be unique, or effectively unique. So if we do the same type of FBA on PDH, as we go from 95% of the max growth to the maximum growth rate, and run it in the me model, this is going to kind of condense to a single point at the maximum growth rate. So we're going to get one prediction of the flux through that reaction and all the fluxes throughout the network. Another thing that's very interesting about me models is they provide a multi-scale type of context for every simulation. So just like an M model simulation, we're obviously going to have predictions of all the different fluxes through all the different reactions, which is something that can be validated from different types of fluxomics data, and the me model captures those predictions pretty well. But unlike M models, we're also going to have to predict the proteome that actually optimizes the metabolic flux that's seen here, so that the proteome optimally produce the proteome to catalyze all those different reactions. And so you can compare this with proteomics data based on the mole fraction of each of the different proteins, and it agrees, at least when you group them into COG categories, pretty well. This demand on proteins is going to ultimately have an effect on the biomass composition, so the rate that each of these different amino acids are produced. So this red bar here shows the prediction, or the fractional abundance of each amino acid when grown on galactose, this teal line when grown on fumarate, and the purple line, which is just the coefficient from the biomass objective function. And you can see that, number one, it agrees with the M model pretty well, at least in general terms. And you can also see that there's a difference between the biomass composition if it's grown on galactose or fumarate. So this is now the different biomass fluxes are now a property of each of the of the growth conditions. So then, to complete this circle, the biomass composition, or the optimal biomass composition, is going to inform then the metabolic fluxes throughout the system. So in order for just one me model to solve, you have to reconcile the organism on all these different levels, which can all be kind of compared to experimental data. So now I'm going to talk a little bit about the limitations of a me model. The most obvious one is that they have a fairly large parameter space. So we have parameters that are similar to the M model, which are the growth-associated and non-growth-associated maintenance terms, or the ATP demand terms, which basically accounts for all the ATP that's not modeled when considering the cost of the cell when growing. But in addition to this, we have kind of the similar idea that's applied to protein. Yeah, so this first gamma and eigenvalue, they're going to be reduced because a lot of these ATP processes are actually modeled in the process of synthesizing protein and RNA. So they're going to be reduced from the model. But we have this extra term, which we call the unmodeled protein fraction, which basically accounts for all the protein that either aren't required for a model to solve due to like gaps in our knowledge, or because they just aren't included in the model at all. And in this case, it's set at about 35%. But the real parameter that gives us the most headache is this K-effective vector. So basically the efficiency of each enzyme, metabolic enzyme, to catalyze its reaction or function. So this is a vector of about 1 by about 5,000. So it's a difficult parameter space to try to really understand, because in order to sample that solution space, we're going to need millions and millions of computational time, which we don't really have. But in addition to being large and difficult to study, they're very important for me-model simulations. And this is because me-models, basically they're not kinetic models in the original sense, they're not ODE models, but they do include those which are pseudo-kinetic terms. And they're ultimately pretty important for defining the flux solution and the way that flux is allocated in a single simulation. So yeah, these can be thought of as kind of cost-benefit models, where the size of the protein and all its cofactors is kind of a cost, where the K-effective is the benefit of actually making that protein. Yeah, so this generally provides the econometrics of growth. So as a result of these kinetic terms, incorrect parameterization or incorrect K-effective values can lead to incorrect predictions of gene expression. So if we go back to this PDH example here, this protein, like I said, is very expensive. It's one of the most expensive proteins in the model. It's about, like I said, 60 subunits. So that's a lot of reactions that have to be active at very high rates in order for this protein to be expressed really at all. But as a consequence of this, it does some things like substrate channeling, and it's very optimized to catalyze flux just insanely efficiently. So as a result, this reaction, it's very, this PDH K-effective for this enzyme, it's very important that this is set to a properly high value, a high rate, in order to reflect the efficiency of the enzyme. This is just kind of one example of why these K-effectives on a systems level are actually really important. And so kind of the first me models that were developed, we centered all the K-effectives in the network around 65, which is kind of a very rough approximation of the average K-effective for a metabolic reaction. And then we kind of scaled it based on the size, based on the solvent accessible surface area of each of the proteins. And so this really does not accurately reflect the biology of the system. So it's not a great assumption. So we started looking into this a little bit further and trying to optimize the way we actually parameterize the model. And the first approach has been this kind of bottom-up way of doing this, which has been a student in the lab has come up with a machine learning pipeline that basically uses fundamental features of both the network, the structure of each of the proteins, so some of the things like the active site properties, the size, the prosthetic groups, and so on, the biochemistry of each of the different reactions. So if it's a dehydrogenase, it's going to have probably different kinetics generally than like an isomerase. So we wanted to include that as a feature in this prediction pipeline. And then assay conditions like pH and temperature. And so we took all these different features and trained it against K-cats that have been measured, just K-cats on Brenda, so they've been measured in vitro. And then also predictions of K at max, which is predicted based on Fluxomics and Proteomics data. So as a result of doing this is basically when we take the results of that machine learning pipeline and plug them into the me model, we get a correlation of protein between simulated and measured value that actually looks pretty good. It recaptures basically general mole fraction allocation of each of these proteins pretty well for each of the each individual metabolic protein, so we're pretty happy with that. But then we also want to,
You go back to the previous slide, it looks like some of them were much more accurate than others, like the light blue one seems to be like really bad, right? And could that just be skewing the overall score, right? Like, have you kind of dug into it and seen like which ones are good and which ones are bad?
So we have a little bit, kind of trying to pinpoint all the outliers and figure out what's causing that. The issue here is, you're pointing out these down here, is this is a log scale. So the mole fraction of those different, the investment in those different processes are just so low to begin with that very tiny changes can change this expression from like down here at 10 to the minus 6 up to the basically where it needs to be. So it's very hard to pinpoint what specifically could be causing those to be outliers, because it could be the model under predicting the reactions that enzymes actually involved in, so the rate that those reactions occur. It could be those very small flux inefficient reactions are a little bit harder to predict or measure in the first place. So they really don't, they're going to be tricky to try to address on an individual level. But most of these enzymes that are more highly expressed usually fall within an order of magnitude of what's measured, and those are the ones that we've kind of focused on because they really drive the phenotype more as a whole compared to these small low activity reactions. So the second approach we took is a kind of data-driven, proteomics-based approach to try to go through proteomics data and pinpoint which K-effectives are very important for general proteomics agreement with the data that we have. So this is going to pick out cases like that pyruvate dehydrogenase, and it's going to say, hey, this reaction is very important that is parameterized correctly, and the efficiency of that protein is accurately reflected. Otherwise, it's going to throw off the simulation as a whole. So that's kind of the logic behind this type of approach. And what this did is, for 4 different proteomics data sets on different glucose conditions, it used this iterative K-effective algorithm where it took in the proteomics data, ran me model simulations, computed the flux per protein, and then tried to minimize the distance between the predicted proteomics and the measured proteomics. And it came up with a K-effective set for each condition that had the best agreement. And then to really figure out which of those K-effectives are the driving factors and getting that good agreement, we then took each of the K-effectives that came out of step 1 and sampled the starting vector and then reran it. We sampled the starting vector and then reran the me simulations to see how it compares with the proteomics data. And then as a result of that, we could figure out which proteins across all the different data sets actually had the biggest effect on either making the prediction with proteomics better, or data better or worse. And then based on the agreement between all 4 proteomics data sets, we could get a set of universal parameters. So these are like kind of universally high confidence K-effective parameters. So as a result of this, there ended up being 284 of around the 5,000 metabolic K-effectives that were deemed to be very significant and important for getting good me model predictions. And these are the 284 K-effectives that were deemed to be important in all 4 of these different carbon sources. And so if we actually take these K-effectives and use them in the model, we see that for, so this is differential expression. You can't really see the left bar, but it's wild type or just glucose grown, the accuracy of just glucose and that substrate, and the differential, The left bar is without the 284K effectives, and the right bar is with the 284K effectives. And this is showing the agreement between model predictions and measured proteomics in response to glucose supplemented with these 4 different 3 amino acids and 1 nucleotide. So when we use these parameters, we can better predict things like differential expression of mRNA. And so these are basically making the model better, and we were able to show that. So this then leads us to believe that these specific sets of K-effectives will allow us to capture global trends better using the me model. So in summary, me models explicitly compute the optimal proteome allocation required to sustain a metabolic flux phenotype or metabolic flux state, and that's really what makes them interesting or why we want to study them. And they do this by first including the model reactions to explicitly model transcription, translation, complex formation, as well as the metabolic reactions in the M model. And in order to basically link all these different major processes together on different timescales, you have to develop coupling constraints to tie the macromolecules created by these processes to the reactions that they actually catalyze, which is a very important thing to get right, as we showed. And then the third step is we have to make sure that the simulations of the model are actually mass balanced. So we need to find a way through the kind of redefined biomass objective function to restrict the biomass created from all these processes to equal the basically rate of dilution or the growth rate of the organism as a whole. So this is going to be analogous to the model biomass objective function. And that's pretty much all I have. So if you guys have any questions, feel free to ask.